Yes. Hello students. In our last class, we were discussing about the muscle. I mean how the muscles are going to manufacture, how their mode of arrangement. So I can quickly revise the things. See, when we have muscle, that muscle is made up of muscle bundle. Yes. So definitely the muscle bundles are held together by. So I mean to say, in one muscle we'll be having many muscle bundles will be present. So in that, each and every muscle bundles are held together by the collagenous protein, so called as fascicles. So in that, one is considered as fasci, many together called as fascicles. So here again the muscle bundles are made up of muscle fibers. They are made of muscle fibers. So again the muscle fibers are made of myofibrils or myofilament. They are made of myofibrils or myofilament. So these are the mode of arrangement of the muscles. Again in this, when we have, when we take the myofibril or myofilament, in that, so the outermost layer, nothing but the plasma membrane is called as, I told you yesterday, sarcolemma. Yes or no? So again, cytoplasm is called as sarcoplasm. Endoplasmic reticulum called as, that is sarcoplasmic reticulum. And the sarcoplasmic reticulum or the endoplasmic reticulum is the very good reservoir that can store the calcium ions. Remember, so it can store calcium ions. How and when? So I can get it later, no doubt. But here we need to understand the sarcoplasmic reticulum. They can store the calcium ions. This is what? In that, when we take the one simple, that is one muscle bundle, here we will be having what? A muscle unit. So that muscle unit is considered as sarcomere. Remember, sarcomere refers to one muscle unit. One muscle unit is called as sarcomere. That muscle unit can be starts with that is Z line, 2 Z line. Remember, yes sir. Likewise, we will be having many node. Again, from this Z line to this Z line is considered as one sarcomere or one muscle unit. Again, from this Z line to one more Z line. Again, second sarcomere or second muscle unit. And in one sarcomere, again we will be having what? A kind of protein. That kind of protein is nothing but that is what? This is one, and again we will be having second protein. Again we will be having one more protein. Yes, remember. So this is A, and we will be having I. Nothing but this one is considered as I and this is considered as A. Refers to actin and myosin. That is thick and thin filament. We will discuss it later. Again, here we have one more proteins. Yes, this is what the structure. How the actin and the myosins are going to arrange within the sarcomere. Yes. So during contraction and relaxation, as we know that the muscles have the capacity to undergo contraction and relaxation. I mean to say that like this. So whatever you do, if you walk or you can run, you can do any kind of work, no doubt, there must be a contraction and relaxation of muscles takes place. So during the contraction and relaxation of muscles, so these actin and myosins are undergo contraction and relaxation. So this is what, and this is completely based on one proposal which is given by the H. Huxley called as sliding filament theory. That we'll discuss it later. And before entering into the sliding filament theory, that is how the contraction and relaxation takes place in the muscle units, that is in the sarcomere. Before entering into that, what is the actual structure of this actin and this myosin? Yes or no? So that we'll discuss in today's class. First we have actin. How the structure will be present. See how the actin is made of. Simple. First we will be having a DNA-like structure. That is, you can call it as F-actin. Just remember, this is considered as F-actin. It is made up of the globular proteins. Just remember, it is made up of F-actin. The F-actin is a protein which is made up of globular proteins. Again, this line again made up of some globules, like this. So globular proteins. So those globular proteins are called as F-actin. Globular proteins are called as F-actin, and this is a thread-like structure. Here, can you see this? The thread-like structure is considered as tropomyosin. It's considered as tropomyosin. Remember, so this tropomyosin is a protein which can run continuously, along with the F-actin, that is globular proteins. These round-like structures are considered as F-actin or globular protein. Along with the globular protein, we will be having a long chain is considered as tropomyosin. And very important thing is, on the tropomyosin, not continuously at a regular interval or discontinuous, there are some structures like this. There are some structures. They are considered as troponin. Remember the word troponin. So the word troponin refers to some of the protein structures which are present on the tropomyosin at irregular or regular interval, not continuously. Remember, not continuously. So they are present here, somewhere on the tropomyosin. Yes. Now it is considered as structure of the actin. So what is the role of tropomyosin? What is the role of troponin? What is the role of F-actin? So we'll discuss while discussing about the sliding filament theory. This is what the structure of actin. Just remember. And the second one is myosin. Remember. So what is the structure of myosin? Here exactly look like a snake. This is what the structure of myosin. Here, this is considered as a head part and is considered as tail part. And again, we are considered it as a arm. And here, we will be having what? 2 out of 4. Totally 1, 2, 3, 4. Out of these 4, 2 are considered as do remember, out of 4 structures, I mean to say, in the they are like a snake. Yes or no? In this, in one head, there are 2 structures. Again in this, again 2 structures. 2 plus 2, again we will be having 4 different structures. In that, out of 4, one from this one, another from this one. Together 2 are nothing but actin binding site. Just remember, actin binding site. So here, actin binding site refers to this structure is going to bind at this particular place, so called as actin binding site. So that too, that's a troponin. Remember, see, during the contraction and relaxation, this actin is going to bind to the head of that myosin. So particularly, it is going to hold the troponin. The troponin part is going to hold this one, that is actin binding site. And second one, out of 4, 2 are actin binding site and remaining 2 are ATP binding site. ATP binding site. Why? Because during sliding filament theory or during contraction and relaxation, so regularly or continuously they are going to use ATP molecules. Do remember, it's the most important thing. Regularly or continuously they are going to use ATP molecules. Hence, we need ATP. So that ATP is going to bind to the head part of the myosin. And this whole structure, that is myosin, is made up of H, M, refers to, just remember the word. Here the word HMM refers to heavy meromyocyte. Remember the word H refers to heavy meromyosin, and here we can call it as light meromyosin. So this is what the part of the this one, the myosin structure. Just remember these two structures, and when we need to understand, or when we understood these two structures, definitely we can understand the sliding filament theory, because that whole sliding filament theory is based on these two structures, that is actin and filament. Clear? Now we can move on to the sliding filament theory. Sliding filament theory. That is, this theory is proposed by H. Huxley. Remember the scientist? H. Huxley is the first person who proposed sliding filament theory. Refers to the sliding of the muscles. The sliding of muscles, nothing but the thin filament over thick filament. Yes, thin filament over thick filament. See in this, who is thick filament and which is the thin filament? Here, when we write actin and we write myosin, in that actin is nothing but thin filament and myosin is a thick filament. Remember, actin is a thin filament, myosin is a thick filament. Thick and thin filaments are present. See here, according to my statement, sliding filament theory refers to the thin filament can overlap or it can slide over the thick filament. So this is what, as we see, just imagine it is considered as muscle. Very important to understand. Here we have muscle. So on the muscle membrane we will be having the snake-like structure. Yes, is called as myosin. Yes, is considered as myosin filament. Myosin filament. Do remember, first we have muscle. Yes, I mean to say, in this example I am taking the muscle which is undergoing contraction or relaxation. Simple. See, imagine it is at resting stage, and here it is considered as at resting stage diagrammatic representation. Yes, out of these four, two are actin filament, actin binding site, and two are myosin filament, two are ATP binding sites, and it is at rest condition. Remember, at rest. See, just imagine at rest it is at 110 degree Celsius. 10 degree. The angle will be 110 degree. Just imagine. And the same time, when you start, see, in this diagrammatic representation, when you start contracting any of the organ in the body, so immediately what happens? So from our central nervous system, remember the word from central nervous system, the motor neuron. We have from the central nervous system, definitely there will be a sensory neuron, means a neuron which carry the information from brain to a particular organ or particular tissue or particular cell. So at the same time, during contraction and relaxation of any part of the body, immediately the brain is going to send. The word brain refers to central nervous system. From there we are going to get sensory nerve, and this sensory nerve is going to bring, that is, it can carry the information to the muscles. When it can reach the muscle, then we can call it as Neuromuscular Junction. Please remember the word. Yes, no doubt it is motor neuron. Yes, Neuromuscular Junction refers to as the name itself indicating the meaning, neuro refers to neuron, muscular refers to muscle, junction refers to where there is a joining or where there is a junction between that neuron and the muscle is considered as neuromuscular junction. Yes. So before understanding this concept, I would like to tell you the thing. Before sending this information from the CNS to muscle, first we need to understand. We need to send the information to the central nervous system. Yes or no? Yes. Here this particular thumb is in the resting state. So now I start contracting. Yes or no? If I start contracting means first I need to send the information. That information is in the form of stimulus. Remember the word stimulus. So any environmental changes are simply considered as stimulus or stimulation. So that stimulation can carry the information from particular organ to brain in the form of, that is, sensory neuron. It's considered as sensory neuron because in our body there are two types of neurons or three types. So fundamentally there are two types. First one is sensory neurons. The word sensory refers to those neurons can carry the information from a particular organ or particular tissue or particular cells to brain. At the same time, the brain can process all these stimulations and resulting it is going to send an information to the particular or respective organ called as motor neuron. Refers to those neurons can carry the information from brain to a particular target organ called as motor neuron, and some of the neurons can carry the information from tissue to brain as well as from brain to tissue are simply considered as mixed neurons. They are limited in number, but anyhow leave it. Here during the sliding filament theory, according to the person called as H. Oxley. So first of all, the muscle is going to send an information or it can send a stimulus from the particular muscle to brain. We can call it as central nervous system in the form of sensory neuron. Yes. As a reward, so the central nervous system is going to send a message in the form of motor neuron to the particular organ, that is from CNS to particular organ called as, that is muscle. Now there must be a called as neuromuscular junction. Yes. Now this neuromuscular junction, just remember very important words are here, and this soon after this neuromuscular junction, a particular this part is going, that is neuromuscular junction is going to release a neurotransmitter. The word you can call it as, you can call it as neurotransmitter. Do remember. So the word neurotransmitter means it is a kind of chemical that can carry the information from one neuron to the next neuron. So called as neurotransmitter. In our concept, the neurotransmitter refers to acetylcholine. Remember the word acetylcholine. So that acetylcholine is nothing but neurotransmitter. When this neurotransmitter is going to release into the muscle refers to sarcoplasm. Remember the word sarcoplasm. See, getting my point? First, we need to send information to the CNS in the form of sensory neuron. Leave it. As a reverse, the central nervous system is going to give or it can send an information to the muscle in the form of motor neuron to the muscle. Now, the junction between neuron and muscle is called as neuromuscular junction. Yes? That neuromuscular junction is in the form of neurotransmitter. That neurotransmitter refers to acetylcholine. That is a chemical that acts as a neurotransmitter. So apart from this, we will be having heparin. Apart from this, we will be having dopamine. Apart from this, we will be having many chemicals are present in our body. So they are considered as chemical messengers. Now, so this acetylcholine is going to release into the sarcoplasm. So as the acetylcholine is going to increase in its amount in the cytoplasm or in sarcoplasm, resulting in the sarcoplasmic reticulum. Remember the word sarcoplasmic reticulum. That sarcoplasmic reticulum come into action and it starts producing the calcium ions. Please do remember this point. Yes, immediately this starts producing the calcium ions. As I told you in earlier classes, I mean to say just before this, sarcoplasmic reticulum is a reservoir of calcium ions. Yes or no? So for that reason, the sarcoplasmic reticulum starts producing the calcium in presence of acetylcholine as a neurotransmitter. What happens? What is the role of calcium here? Definitely we need to understand. Yes. Now these all calcium ions can release into the muscle, and here, yes, just remember, here you will be having what? Can you tell me what is this? Actin diagrammatic representation. Yes or no? Yes, actin. So the actin is made up of three parts. One is the round globular structures called as F-actin, and the long continuous thread-like called as tropomyosin, and at regular intervals we will be having small structures called as troponin. I told you people, troponin. So yes, what I am trying to tell you is that in this concept, when these two are at a resting condition, here these troponins are masked. Remember the word. These are always masked. Here the word masked refers to they are always closed. They are always closed. It refers to they never ever attach to these two sites called as actin binding site. I told you people, actin binding site. So because this actin has to bind with the actin binding site, but it is masked. So because of this condition, these calcium ions starts and go and bind to this actin binding site. Here we will be having actin binding site. So when these go and attach to the actin binding site, and they can remove the mask. They are going to remove the mask from the myosin as well from the actin. Now what happens? Ultimately, when the calcium are going to remove the mask, definitely what happens? This actin go and bind to the like this. Clear my point? I hope you understand this concept. Now there must be a cross bridge. You can call it as cross bridge. So the word cross bridge refers to this actin binds to the myosin at actin binding site is considered as cross bridge. I hope you understand. Yes. Now this is over. And now what happens? Immediately the ATP is going to release adenosine triphosphate. So now this ATP is going to utilize at particularly this point, resulting in the ATP can give rise to ADP plus inorganic phosphate. Remember, I mean to say what I am going to do here, this ATP is going to utilize by the actin and myosin, resulting in the ATP can break into ADP, that is adenosine diphosphate, from triphosphate, adenosine triphosphate to adenosine diphosphate, and inorganic phosphate is going to release. Clear? I mean to say here the most important thing is the ATP is going to utilize for the cross bridge. This is what is in the next step. Yes. See, till now just we discussed the thing, that is myosin filament hold the actin filament over. That's all. Next, in the next process, same, that is we have a muscle bundle. On the muscle bundle we will be having that is a snake-like structure called as myosin. The myosin will be having 4 different structures called as actin binding site and ATP binding site. See, as I told you people, the actin is going to hold the actin binding site of the myosin, and here you can call it as a cross bridge. Now, at the same time, according to the sliding filament theory definition, so the thin filament can that is make a cross bridge on the thick filament. Yes or no? So the crossing between, I mean there must be a binding with the actin as well as myosin. In the same structure, here we will be having the actin. So this actin starts moving, or it can start pulling this head part. Now, here I told you people, imagine 110 degrees Celsius. Now it will become 90 degree. Yes sir, the angle will be 90 degree. And in the next step, again it will be same. That is, muscle we will be having. Yes, now you can understand by the diagrammatic representation. Now the angle become 90 to 60. Means again one more diagram, it will become. Yes, you can understand. It will become 30. Means, see, in the first diagrammatic representation, here it will be 110 degrees. The angle will be 110 degrees refers to it is completely at rest. So the whole muscle is at rest. When there must be a cross bridge takes place, means the actin starts pulling the myosin head part, or you can call it as HMM or LMM. Nothing but heavy meromyocytes or light meromyocytes. Now the angle become 90. The angle become 90 means undergo contraction. Yes sir, undergo contraction. Here 90 becomes 60. Again further contraction. 60 become 30. Again maximum contraction. Maximum contraction. Yes. So at what point? At what particular point? The 30 refers to maximum contraction takes place, means all ADP are going to utilize, resulting in the again the actin binding site become mask. Yes, again actin binding site become mask means again there is no relation between the actin and the myosin. So for that reason, again ultimately the same diagrammatic representation takes place. Yes, means resulting in the again the muscle will come back to its original position. This is nothing but sliding filament theory. So the thin filament can slide over the thick filament. This is what our today's concept. You can call it as sliding filament theory, which is given proposed by a person called as H. Huxley. Yes, I think it is enough for today's class, and remaining things we'll discuss in tomorrow's class. Thank you.